Hi dear students, welcome to one more session of genetics. There is an old saying that like begets like, and we can see that almost every offspring shows some resemblance to their ancestors as well as their parents. Let it be a dog, let it be a cat, let it be a bird, or let it be a human being. Irrespective of different species, you can see that they show resemblance to their parents. And this resemblance is referred to as the heredity. The process of heredity is considered to be a mechanism to conserve the parental characters. But at the same time, we can find that there do exist some variations or some differences as the generations are being derived. And that has led to the new science referred to as genetics, which we study right now. In the coming sessions, we will be talking about the classical genetics, especially giving more relevance to the Mendelian genetics. The early attempts to prove heredity was being carried out by various scientists, and we could see that promising experiments were being conducted even in 1799 by Knight, and he was working on edible pea, and an observation that he was quite interested was that when an unpigmented pea was pollinated with a pigmented pea, it gave rise to only pigmented pea. And when this pea was self-pollinated, it gave rise to both pigmented and unpigmented varieties. Knight was quite interested in it, and he was also fascinated by his observation, but he could not give any mechanism underlying this phenomenon. Now, similar experiments were also being conducted in 1763 by Carl Render on the tobacco plants such as a nicotiana. You know, nicotiana, that is a scientific name of tobacco. So, different people had conducted different experiments regarding similarity between the offsprings, but they couldn't explain what was the reason for this similarity or this heredity. And a key to the mechanism of heredity was developed by none other than Gregor Johann Mendel, who is regarded as the father of genetics. The modern science of heredity starts with the experiments of Mendel on the garden pea Pisum sativa. Now, who is he? Mendel was born in 1822 near Brunn in Austria in a family of a poor farmer. And as a son of a farmer and a fruit grower, Mendel showed a very keen interest in plants and animals, and even from childhood, he was helping his father in the garden, as well as he was quite interested in the plant hybridization techniques. So, as he grew up, what happens after completing his 2 year university course in philosophy? What he did, he entered an Augustinian monastery and was ordained as a priest at the age of 25. Now, after few years, he was sent to the University of Vienna to take up a formal coursework in physics, mathematics, and natural science. He was not a very good, outstanding student at the university, but the university experience was quite interesting, and it induced further interest in scientific and experimental work in him. And what happened in 1854, after completing his study, he was appointed as a substitute science teacher. And it is during this time that the genius in him was evoked, and he conducted lots of experiments on the pea plants in the monastery garden. And it was the famous experiments of Mendel which laid down the foundation of genetics. After 7 years of painstaking, sincere, and devoted experimentation, he in fact presented his findings before the Natural History of Society of Brunn at two of its meetings in February as well as in March 1865. And this paper, entitled Experiments on Plant Hybridization, was presented in German language and was also published at the annual proceedings of the society in 1886. But Mendel died in 1884, and his work was not even then recognized during that time. And it was later on in 1990s a rediscovery of Mendel's work was being done. It was mainly when three scientists, De Vries of Holland, Correns of Germany, and Semak of Austria, they also found similar results. And after this discovery, there was a spurt of interest in Mendel's findings, and the science of genetics was truly born. Although the principle of genetics was enunciated in 1865, the newborn was kept in an incubator and forgotten for 35 years. So that's about Mendel. Now, what has he done so far to be called as a father of genetics? Let's explore that. Now, let me just take you to the animations provided by the Cold Spring Harbor Laboratory, my favorite spot to explain genetics animations. And now here let's see Gregor Mendel and his work. Let me introduce him as they have explained here. He worked with pea plants because they were easy to grow and they have many traits that could distinguish strains of pea plants from each other. And the traits could also be tracked from one generation to the next. Now, when left alone, you can see that the pea plants would self-fertilize. If you go to consider the immature flower, you can see that there is a pistil as well as a stamen here. This is the stamen, and the stamens, they are the male sex parts, and they mature first and drop the pollen inside the immature flower. And the pistil, the female sex part, it usually matures later and its eggs are fertilized by the pollen that lands on the pistil. So, in order to cross-fertilize the pea plants, Mendel opened the immature flower and he cut off the stamens before they could mature and drop the pollen. And after the flower matured, he dusted its pistil with the pollen from another plant, and this is called the cross fertilization. So this was some of the experiments or the basis of the experiments which were being conducted by Mendel. Now let's see why he chose this and what are the parameters he chose or what are the traits that he explored. Rather than looking at the pea plant as a whole, Mendel he focused on 7 individual traits that could be easily distinguishable. He found that each trait had two alternate forms. Now, for example, if you go to look into the seed color, it could either occur as a green pea or it could occur as a yellow one. And by analyzing the results of various crosses, Mendel concluded that each alternative form of a trait is specified by an alternate form of a gene. And if you go to look into the flower position, they could be either be terminal or they could be axial, and the flower color should be either purple or it could be white, or the plant height could be either tall or it could be dwarf, and the spot shape could either be inflated or it could be constricted, and the pod color also varied from yellow to green. And what he did, Mendel chose pea plant mainly because the pea plant was something which you could, uh, which naturally did self-fertilizing, but at the same time, it could be a multi or it could be what it could be, instead of pollinating it, he could also artificially pollinate it. And he found that they were pure bred strains. That is, the pea plant was purely bred, and such purely bred strains were also being found to be available. And each strain had only one form of the gene which determined the trait. So let's just explore what was his experiments. Now you saw what is the experimental tool and why he took it. And now let's explore how he did it. Before going into the details, let's just see a short animation here where Mendel is explaining the pea varieties, and he saw that peas have distinctive traits that are inherited in predictable way. And the visible trait is often referred to as the phenotype. And if you go to look into the plant, these there are some of the traits which are visible, or the phenotypic traits of pea plant were being considered. And in the stem and the flower traits, he located the flower position, and which was being found to be the top as well as the side one, and the stem length as we expressed earlier, the tall and the dwarf, the pod traits, the pod shape, as well as the pod color, then the seed shape, the seed color, and the seed coat color. And these are the seven traits which he had taken, and each trait had two phenotypes. What are the phenotypes? Phenotypes refers to the visible effect of, or the visibly available trait. And instead of taking all the traits, now we are just considering some traits here, the seed color, the green and the yellow. And what Mendel did is he purchased some purebred seeds for his experiments. And usually it has been considered that during every harvest, some of the farmers will keep some of their pea crops as seeds for the next year. And what happened? These seeds came from the pea plant's regular habit of self fertilization, and that's how he got the purebred strains. And in the case of seed color, one strain had green seeds and the other had the yellow phenotype. And if you go to consider that the phenotype is being responsible by the presence of certain genetic factors, and it has been found that the genotype of a green type of seed could be of and having a small alleles, that is small y and small y, as well as the yellow seed had alleles capital Y and capital Y. And what is an allele? Each form of the particular gene is referred to as a allele, and the pair of allele is referred to as a genotype. The purebred peas have two copies of the same allele. I guess now you got an idea of what is phenotype and what is genotype, and why Mendel had selected the pea as his experimental plant.
Now that he knew the principles of artificial fertilization, what Gregor did was he crossed a true breeding parents of purple flowers with white flowers, and he resulted in the formation of hybrids, and he found that all the plants had purple flowers. And when the F1 generation was being allowed to self fertilize, he found that there was a mixture of flowers, like some of them were being found to be purple flowers and some of them were being considered to be as white flowers. Moreover, there was also a strict ratio which was being followed, giving a 3 is to 1 ratio in the F2 generation. So let's see what were the other features also that he observed. He also noticed that when a puffed pod was being crossed with a pinched pod, it only remained puffed. And if you go to cross a yellow seed with a green seed, it was yellow. A tall plant with a short plant, it was always tall. And he needed to find out what was the reason of this. And if a side flower and top flower was being crossed, only side flowers were being formed, and he needed to find now what was the reason of this. Now, if you go to look, the colored coat was being coated with a white coat, that is also only a colored coat. And a round against a wrinkled seed, you always arrived a round seed in the F1 generation. Thus, he had to see that when even when the green and yellow is using, the green is only being formed. Now, what was a particular reason for this? It indicated that all this, all the offsprings which are being formed from the purebred parents, the traits were not mixing with each other, or they were not blending with each other. And if you go to look into the yellow green, yellow versus green, never the hybrid was, the F1 hybrid was not found to be yellow green, and a tall with a short was never to be average height, and that's what he concluded. And further on, he considered the F2 generations also, and he came across with some of the laws of Mendel, and we will see that in the coming sessions. Thank you for now.